one of the most fascinating topics ever is that of electric signaling in biology. It's just unbelievable how we are constantly relying on electricity for our bodies to function like little robots, and not just the brain or nerves, but essentially every cell is relying on the mechanics of electromagnetism. It is so cool. But first, it's important to demystify the term electricity. Most biologists aren't really aware of what it really means. It just seems like a cool, powerful energy source for your phone or computer, or you may picture a little spark or a lightning bolt. But in reality, electricity is just the movement of charges. Charges are, of course, atoms with an unequal amount of electrons and protons, resulting in either a positive or a negative charge. When something has a charge, it usually has a desire to lose that charge, either by gaining an electron or by losing one. To do this, some type of movement is involved. So you will usually see a negative ion moving towards a positive ion to exchange one electron. Actually, to get more specific, movement and energy are super closely related. So the transfer of energy, or even energy itself, they are inseparable from the motion of objects or particles, which is also super interesting, and I will make another video for that soon, but just keep in mind that motion and energy, and hence electric energy, are best friends. So when something has a charge, it will move towards oppositely charged entities and away from like charges. When the charges are moving, this is called a current, exactly like a water current. You have all these particles in here, and as they are moving, you create a current, which is denoted by the letter I. And more formally, it's defined as the amount of ions that cross a certain area per time. And that's exactly it. That's electricity. That's all it is. It's just the movement of these charged particles called ions. So electricity in the body is not a spark or battery, it's just positive or negative molecules moving around. In the body, every cell has a membrane potential. This means that ions like potassium, sodium, chloride, and calcium are distributed unevenly across the membrane. Like in this example, there's way more calcium on the outside of the cell than on the inside. In this case, there's a chemical potential energy. This means that calcium wants to move from an area where it is highly concentrated into an area where it is not. This is the same principle as osmosis, and it's actually hinged on enthalpy and entropy, which is also super interesting, but a bit out of the scope for most biologists. But if you're interested in that connection, hang around to the end of the video to get some more insight into that. But the essential idea is that every ion has a concentration gradient. So at this point, there is a high concentration of calcium, whereas here there isn't. So the calcium experiences a driving force that's moving it to the inside of the cell, where it can move down this gradient. And of course, remember that this is relative. It's all about the difference between these two areas. When these calcium ions are close together, they experience a lot of intermolecular forces. So this is like van der Waals forces and dipole-dipole, depending on what kind of molecule you have. But this means that there are attractive or repulsive forces between the ions. In this case, because they have the same charge, they repulse each other and want to get away from each other. This means that as these molecules are moving further apart from each other and becoming more sparse, like in this environment, there is less of this repulsive force, which means that each of the calcium ions experiences a lower energy state. And you will notice that in chemistry, almost everything is trying to lower its energy. Pretty much all chemical behavior and interactions are explained by this. There are a lot of ions in the body, and they all have their own concentration gradients across membranes. And that also depends on what type of cell it is, or where it is located, or what kind of channels it has in the membrane that allow the movement of these ions. But a lot of processes are based on the movement of these ions, aka changes in the membrane potential. And this movement of ions is, as you remember, electricity. Most people know that nerves function based on this principle, but it is necessary everywhere. For example, getting glucose to enter the intestinal cells during digestion requires a concentration gradient of sodium. There's a higher concentration of sodium inside the intestines than in the cells that line the intestinal walls. Hence, sodium experiences a desire to enter these cells. However, it cannot do this without glucose. As it is trying to move through these specific sodium channels, it is hindered by the channel, because the channel only functions if glucose also enters. Channels in the membrane wall are made of protein and can be quite complex in their configuration, aka their shape. So they can actually select very specifically what kind of molecules or ions can pass through them and in what situations. So in this example, this sodium channel will only open up if it has both sodium and glucose attached to it. So although glucose may not want to enter the cell, it has an affinity for the channel and will attach to it eventually. So then when sodium also enters the channel, it is moving down its concentration gradient, aka losing energy. And this energy is being used by the channel to change configuration to allow the glucose to enter the cell. It's basically holding on to both the glucose and sodium until it gets the energy to change its shape and release them into the cell. So without a sodium gradient, we would need a totally different way to move glucose into our body. If you're more into biochemistry, the production of ATP by ATPase is of course dependent on the proton gradient across the mitochondrial wall that powers the joining of the third phosphate to ADP. So it's true, every cell is deathly dependent on gradients. Interesting is also the example of yeast, which functions through anaerobic respiration, aka fermentation, to produce alcohol. Alcohol is reactive enough to steal the ions across the cell membrane and react with them. So a big problem in industrial winemaking is actually the fact that yeast kills itself by removing its cell's chemical gradients, and hence membrane potential, as it is producing alcohol. And this limits the amount of alcohol a certain amount of yeast can produce. And that's also an economic problem, where industrial chemists can attempt to find ways to get the most out of these poor guys. But in the case of neurons, they utilize rapid changes in the membrane potential by suddenly allowing certain ions to enter the membrane, or not. This influences the nearby environment to also experience the same changes in the ion concentrations on either side of the membrane. As a result, electricity is used as a signal to trigger another neuron, to release a neurotransmitter or a hormone, or to trigger something like a muscle to cause contraction. So yeah, electricity is not some weird, bizarre magic that's happening in our phones or in our head, but it's actually just the movement of charged particles. And somehow, this alone is able to do so many metabolic and amazing things in your body, and we absolutely need it to survive. Bioelectricity is just the best. Or should I say, the bzzzt. Okay, so if you stuck around to the end, it means you're interested in the concept of enthalpy and entropy in the context of chemical gradients. Enthalpy is the energy stored in chemical bonds or intermolecular forces. When like charges are closely together, they experience electrostatic repulsion. This means they have a high enthalpy because there's a lot of energy stored in these intermolecular forces. The ions are now in a greater energy state. As they wish to lower their energy state to get to a more stable configuration, they will move apart to lower the repulsive forces between them. Now, entropy is generally considered a measure of disorder, although more specifically, it's the number of possible microstates a molecule can inhibit, although that will also be explained in a later video. Systems generally want to increase their entropy, meaning they want to disperse more randomly. In a chemical gradient, ions or molecules are more closely together on one side of the membrane than the other. This means as they move to diffuse across the membrane, or just diffuse in general, particles spread out more randomly, and this increases the entropy. In both cases, spreading out decreases the energy states of the particles. This also further explains why it's called a potential. Potential energy is the energy something could expend based on its position. By being closely together, these ions have the ability to spread apart and lower their energy. The movement that occurs while spreading out requires kinetic energy, which is the energy of motion. This kinetic energy can only be taken from the available potential energy. So the potential alludes to the fact that these ions have the ability to move and create kinetic energy. Hence, enthalpy and entropy are driving forces behind chemical gradients. In fact, both of these factors can be combined into a formula to predict if a chemical reaction or behavior will be spontaneous. The factor G is called Gibbs free energy, and if it is below zero, it means that a chemical reaction is favorable and hence spontaneous. And this is more likely to occur if enthalpy decreases and entropy increases, although temperature is also a factor.